Hello everyone, my name is Debbie and I'm a member of the iGEM 2022 team on the web app. This year our project is called Chases. So iGEM stands for International Genetic Engineering Machine. So we basically works with synthetic biology. So what is synthetic biology? SynBio is a design and construction of new bioparts, devices, and systems, and the redesign of existing natural biological systems for useful purposes. Basically, we choose a bacterial chasis, design a circuit with a specific backbone to the species, and then incorporate the circuit to the chasis. SynBio has the potential to solve many real-world problems like food spoilage, algal blooms, plastic pollutions, and emerging pathogens. For example, we can incorporate a circuit to enable B. subtilis or E. coli to degrade microplastics. Despite having so much potentials, without the ways to use those circuits outside of the lab, SynBio application remain very limited. One barrier of making SynBio feasible is chasis selection. As you can see, many factors impact chasis' ability to survive in a deployed site, making it challenging to determine if a given chasis is compatible with certain environments. Traditionally, the way to choose chassis is by literature review, which is reading a bunch of papers on individual chases. But one limit, as you can probably imagine, is that it takes a lot of time, and many papers only examine like one or two chases on like one or two factors, which like pH or temperature instead of like a real environment. Another method to predict chases is by environmental samples. So we go into the field and take 16 S samples and then measure what kind of bacteria exist in that environment. And then depending on whether that bacteria is culturable, we choose to employ it for our circuits or not.
So in our iGEM project this year, we're trying to use data analysis to help researchers with the chassis selection process. The data source we used come from diverse sources, individual studies, online, and also biosample databases such as Akita from UC San Diego, and also field samples. Next slide. And we use different approaches to do the predictions. We use linear regression, nonlinear regression, KNN, random forest, and neural network. We feed the data, we split the data into 3 different sections: training data, validation set, and also testing data. The model are selected with the best performance in response to the validation data. So then we finalize the model and feed into the testing data to see how it fits in the real world. Next slide, please. So the first model we tried is linear regression. So the linear regression that we tried, the result wasn't so good. It wasn't so good, but it gives us a little bit of the insight of how the data actually looks like when we fit it to a data, when we fit it to a model. So, and right on the right hand side is the independent variable by variable plot with the covariance value. Next slide, please. And we also tried a non-linear regression, which we started adding in higher degrees of polynomial and fit into the model. So we reached a pretty good R-square, which is 0.4, which is a little bit better than regression, but we also want to try out some other models. Next slide, please. So we use this supervised machine learning algorithm that is called random forest regression that grows and combines multiple decision trees to create a predictor. So we use this traditional split, which is we made 80% of the data set training set and 20% of the data set validation set. I reached a decent results as for the error metric and also the R-squared. And as we can see on the graph, the algorithm predicts most of the data accurately, but fails to predict some of the outliers. Next slide, please. So we also use this really similar KNN regression from using Python from the sklearn package, and the result is slightly worse than the random forest. As we can see, we fail to predict more data set in this case. Next slide, please. And we also use neural network. We use a TensorFlow package to build our network, which contains 7 layers. So as we can see, we have 1,000 epochs in the very early stage of our training process. The accuracy just skyrocketed. We suspect that it might be a problem with our data source. So more data processing and cleaning need to be done to make our training process better. Yeah. So thank you very much. That is our project.
Thank you.